everyone. In this video, we're going to wrap up our discussion on organic molecules by describing nucleic acids. So nucleic acids are large organic molecules that are made up of carbon, hydrogen, oxygen, and nitrogen, which are all familiar, and then also phosphorus, which is something we haven't seen in the other organic compounds that we've described so far. So nucleic acids generally are used to store and process information inside of cells, and they break down into two classes. Number one is DNA, which stands for deoxyribonucleic acid. Super long name, but we'll see kind of the breakdown of those individual parts of that word in a couple of moments. So DNA contains information to build proteins, and there are tons and tons of proteins in the human body. And so far, we haven't really detailed exactly what their functions are or where they're located or anything like that, but it's coming up very soon. So it's very important to also sort of understand how those proteins come about. So DNA directs the synthesis of proteins, and as a result, it controls all of the physical characteristics of the human body. So everything from eye color, whether you have blue eyes or brown eyes, to how tall you are, to the specific proteins that make up different parts of individual cells. The DNA contains all of that information. It's every piece of the instructions that is contained within a person. And the other class of nucleic acid is RNA, which stands for ribonucleic acid. Ribonucleic acid cooperates with DNA to build proteins. It basically contains a recipe, and it sort of copies the recipe from the DNA and then uses that recipe to actually make the proteins. So when we talk about cells, we'll talk more about the specific relationship between the DNA and the RNA and detail their functions a little bit more clearly. In this video, however, we just want to talk a little bit about the building blocks of nucleic acids and have a little bit better understanding of their basic structure. So there are three important components of nucleic acids. The first one is a nitrogen-containing base, or nitrogenous base, and it literally means that the element nitrogen is one of the atoms that is found in the molecule that makes up this nitrogen, that makes up the base. There are actually 5 different bases, and we'll just draw the structures quickly. And as before with the other organic compounds, of course, don't feel obligated to learn the structures. It's just an opportunity to make this feel less abstract by showing that we have the different elements just sort of organized in specific ways to make up these particular molecules. So first we have adenine, and you can see all the nitrogens that are present. Of course, we know that every intersection is a carbon, and any place where we don't see 4 lines, we know that there are also some hidden hydrogens. The next one is guanine, followed by thymine, followed by cytosine, followed by uracil. Now, it turns out that adenine, guanine, thymine, and cytosine can be found in DNA, and adenine, guanine, cytosine, and uracil can be found in RNA. We also can refer to these different bases just by their letters. So adenine is referred to by A, and guanine by G, thymine by T, cytosine C, and uracil U. So we can just sort of abbreviate by using the letter. The next component of nucleic acids is a pentose, or 5-carbon sugar. So we've only mentioned really glucose in terms of like monosaccharides. We said that monosaccharides could be 3 to 7 carbon atoms, and glucose contains 6. So this one is a little bit different because it contains 5, and we can still see that it's kind of like a ring structure with an oxygen. It's a little bit similar, but it only contains 5 carbons. The particular way that we're drawing this is to sort of give it a little bit of a 3D dimension, so that we can see the relationship of the different atoms within the molecule. But don't feel it's really the same thing. So there are 2 types of pentose sugars that can be found in nucleic acids. Ribose is contained in RNA, and deoxyribose is contained in DNA. So ribonucleic acid contains ribose, and deoxyribonucleic acid contains deoxyribose. And the difference between the two structures is super minimal. It's just one hydrogen is present attached to the second carbon, as opposed to an oxygen and then a hydrogen or an OH group attached to the second carbon. It's a tiny difference, but it's entirely the difference between the sugar component of the DNA or the RNA. And the final component is a phosphate group, and the phosphate group is basically a phosphate with a bunch of oxygens attached to it. And I just want to mention, because later we're going to talk about something called ATP, I just want to mention also that we can have what's called inorganic phosphate. And typically an inorganic phosphate is a phosphate with 4 oxygens, and one of those oxygens has a hydrogen attached to it. So we can abbreviate that PI, and we'll see that a little bit later. All right, so next let's sort of take these components and stick them together and show how we actually can make something like DNA. All right, so first we'll show the relationship between the 3 components. So we have our deoxyribose, and that's attached to the phosphate group. And then we also attach, in this case we'll use adenine, we also attach our nitrogen-containing base. So the nitrogen-containing base and the deoxyribose sugar, or if it was RNA and it was ribose, it would still be the same. The pentose sugar plus the nitrogen base equals what's called a nucleoside. And then when we attach one or more phosphate groups to it, it's actually called a nucleotide. So DNA is made up of a bunch of nucleotides. And the next picture is extremely complicated, but it really just is to sort of show how we can make multiple nucleotides stick together to actually make the DNA. So what we see here, we sort of just rearranged the drawing a little bit, but we can see a combination of, in blue, the deoxyribose sugars and the phosphate groups in sort of an alternating pattern. And then we also can see in red our different bases, and we'll actually label them. And they are, we just are showing one of each, G, C, A, and T. Now, there's a couple things that are interesting about the way that these nucleotides interact with each other. So first of all, the arrangement of DNA is basically like a ladder, and we have the sugar and the phosphate sort of would be like the sides of the ladder, and then the bases, the nitrogen-containing bases, would be like the rungs of the ladder. And the rungs of the ladder are connected to each other by hydrogen bonds. So C and G always pair together. You would never see G and A or G and T. C and G always pair together, and we can see that there are 3 hydrogen bonds between them. And then A and T always bond together, and we can see that there are 2 hydrogen bonds between them. And the sides of the ladder, like we mentioned, the sugar and the phosphate, it's actually called the sugar-phosphate backbone. So just like what we did with the phospholipids, we're going to draw this kind of like with all these crazy molecules one time, and then after that we're going to represent the DNA in sort of a cartoon-like structure. So we'll just sort of redraw it over here and take a look at the cartoon version, which is what we'll draw from now on. So we can actually see the rungs of the ladder are the different bases, and they're opposite. 2 bases are opposite each other, and they pair the same way each time. So we have C and G across from each other, A and T, T and A, and of course in blue the sides of the ladder as well. So technically, DNA is what's called a double helix. So it's twisted in a sort of helical pattern. And so we're sort of drawing it initially just straight to have an understanding of the relationship between the components, but technically it's twisted in this helical shape. And we'll talk more about DNA, like I said before, when we talk about cells. We'll talk about how cells replicate. We'll kind of mention DNA again. All right, so let's quickly compare DNA to the structure of RNA. So while DNA is a ladder, RNA is only a single strand, so it's not double, it's just one sort of side with the different bases, and they don't really attach to anything. And as we mentioned before, RNA uses uracil instead of thymine, so we just have U instead of T. So here's if we were going to draw RNA in the cartoon version, we would just see one side of the backbone, and then we would have our different bases, and we could make any arrangement of them. They're not pairing with anything, but we are going to see, like I said, the U instead of the T. And the RNA is actually made from a DNA template. So we start with DNA and we make the RNA from that, and then we can use the RNA later to make proteins. And that's also something we'll expand upon when we start talking about cells. So that actually brings us to our final point, which is that it's super relevant here to talk about something called high energy compounds. And high energy compounds are basically molecules that contain high energy bonds. So they fall under the organic molecule sort of umbrella because they are made of the same basic elements that RNA is made of. So let's just take a look quickly. We're going to use ATP as our primary example. So if we were to draw the nucleoside for adenosine, we're actually going to draw the adenine base with a ribose sugar. So we're not using deoxyribose, we're using the one for RNA, we're using ribose. And the combination of adenine and ribose makes a nucleoside called adenosine. Adenosine is found in the body and functions in a variety of ways that we'll sort of see later. But it's also a drug that we can administer for cardiac dysrhythmia. So for a patient who has a super fast heart rate, like 180, 200, we can sometimes administer adenosine to that patient. So it's kind of interesting, and we'll see this to be a very common trend, that things that are naturally made in the body are also often administered as drugs in higher concentrations through like the IV route. So it is the same thing. We're just going to draw a comparison here. So if we were to attach one or more phosphates to the adenosine, we can call it a nucleotide, as we mentioned before. But as we're going to talk specifically about high energy compounds, we're going to sort of name it something in particular each time we add a phosphate group. So if we add one phosphate group, we'll call the molecule adenosine monophosphate. And so monophosphate meaning one phosphate, and we'll just sort of observe the pattern here. If we were then to add a second phosphate group, you can see that we'd be attaching the phosphate group to the previous phosphate group. And when we have two next to each other, we actually call that adenosine diphosphate, or two phosphates. And then finally, if we were to attach a third phosphate group, then we would have adenosine triphosphate, or three phosphates. So these three molecules, adenosine monophosphate, diphosphate, and triphosphate, can be abbreviated AMP, ADP, and ATP respectively. All right, so then for a moment, let's talk about why these guys make high energy bonds. So we can see that there are a ton of oxygens that are kind of all near each other, and we know that these oxygens have a ton of electrons that are around them, and electrons are all negatively charged. And some of these oxygens we can see even possess like a true negative charge because they have more electrons than they have. Maybe they'd like to have some more bonds. So if we think back to our prior example where we have two magnets and we take the same ends of the magnet and try to push them together, we know that they naturally want to repel. And so by way of holding them together, we are actually utilizing a lot of energy. So it takes a lot of energy to hold charges together. And so when we think about sticking a phosphate group onto another phosphate group, we are essentially doing the same thing. We're holding a lot of negative charges together, and that results in containing or holding onto a lot of energy in order to make that happen. So that means that the bonds between the phosphates and the oxygens from the third and second phosphate groups contain a lot of energy, and so those bonds are called high energy bonds. And the compound overall is called a high energy compound. So it's actually super important because ATP is the primary energy source in the body. And so when we think about things like saying we need to consume glucose in order to have energy, or we need to, breathe oxygen in order to have energy, ultimately those things are utilized to synthesize or to make ATP, so that ATP can power all of the other activities the body is going to do. So ATP is so important, and it's so important because it contains these two high energy bonds, which means that it's a super excellent energy source for the body. The body can't really utilize glucose and oxygen directly for energy, so it just sort of converts them into ATP and then uses the ATP over and over again. So this is definitely something that we're going to talk about extensively, and it's definitely the most important thing for this particular video. So the nucleic acids, we care a little bit about their structure and try to have a better understanding that's less abstract of nucleic acids, so we can talk intelligently about DNA and RNA and their functions down the road. But ATP is something that we'll talk about constantly, and how everything that the body is going to do that requires energy is going to utilize the ATP. So if there's one thing to remember, this is definitely it. And the last thing I want to mention, sort of in the same family but a little bit just a little bit off topic, is what's called cyclic AMP. So I'm going to again draw adenosine, and we actually have a phosphate group attached, but this time you can see that the phosphate group, instead of just being attached once, it's actually attached twice to the ribose, and it makes sort of a circle between the oxygens and the phosphorus atom. And so this is actually called cyclic AMP, or CAMP, or cyclic adenosine monophosphate. And it's really not classified like a high energy bond, but it is something that we're going to discuss very frequently. And lastly, while we're using adenine as the base for our example here, we could actually replace it with any of the other bases and we would have the same sort of thing. The most common other example would be guanine. So if we use guanine as a base, we could have guanosine nucleoside, we could have guanosine monophosphate, guanosine diphosphate, guanosine triphosphate, and so on. So mostly we'll talk about ATP, a little bit we'll talk about GTP, and a little bit we'll talk about cyclic AMP or cyclic GMP and that sort of thing as well. So that wraps up our discussion of nucleic acids, of ATP for now, and of organic molecules and chemistry. The final bit of information, and now we're officially ready to move on and talk about cells. So I hope you enjoyed it. Thanks for watching.